Okay, so in today's lecture, we will continue this page of your first aid book. Okay, we did kinase, phosphorylase, phosphatase, and dehydrogenase. Okay, and now we are left with four enzymes: hydroxylase, carboxylase, mutase, synthase, or synthetase. Okay. Similarly, we have done few of the rate limiting enzymes yesterday. Up till glycogenolysis, the first 5 ones we have did, and now from today we will see HMP shunt to cholesterol synthesis. Okay, so first of all, let's discuss about these the remaining enzymes we have been left with. Now I will teach you carboxylase first, okay, because in the rate limiting enzymes you will not see any hydroxylase. So I will be teaching you carboxylase and synthase or synthetase first, okay, and then we will study mutase and hydroxylase, okay. So you people should learn and remember the names of the processes along with the enzymes. Okay, so carboxylase, while talking about carboxylase, you see what they've written, that it transfers carbon dioxide group with the help of biotin. Okay, so biotin, what is biotin? Biotin is vitamin B7, right? We have already studied vitamin B7. If I take you back up to B7 and show you, I also told you to remember few of the things. So this is your vitamin B7 biotin. Okay, you see, and also see here it is a cofactor for carboxylation enzyme. So all these enzymes which they have named over here, like pyruvate carboxylase or acetyl CoA carboxylase or propionyl CoA carboxylase, I told you not to remember the names of the enzymes, just remember that B7 is a cofactor for carboxylation enzymes. Okay, so the same thing they have written over here. The same thing they have written over here, that carboxylase transfers this carbon dioxide group with the help of vitamin B7, which is biotin. And they have given the example of one carboxylase, okay, that is pyruvate. Now I'll take you to this page of your Lippincott book, and you see here in this picture, see this picture, this structure you see is biotin. Okay, this structure you see is biotin, and this red structure which is drawn over here is the structure of pyruvate. Okay, now what is going on at this level, that you see what it is written, Car carbon dioxide is activated and transferred by pyruvate carboxylase to its biotin prosthetic group. So, okay, to this group, you see, there was this carbon dioxide is added at this level and it is attached over here. Okay, let me change the color which is not used in here. Let's say, let me take pink. So now you see, this carbon dioxide which was added in this reaction is transferred to this pyruvate carboxylase structure, and this is the structure of biotin. Okay, so it is the enzyme pyruvate carboxylase helped this carbon dioxide to transfer to this biotin structure. Okay, you're getting my point. Exactly the same thing is written in your book. Let's read it once again. Carboxylase transfers carbon dioxide with the help of biotin. So this structure is biotin structure, And with the help of this structure, this carbon dioxide from here is added to this whole structure, okay? And the enzyme which is responsible to make this happen is pyruvate carboxylase. Okay, so I showed you this picture so that you should always just keep this picture, this little reaction in your mind with carboxylases, and you will never forget. Okay, and that is why I showed you the picture, because the name of this vitamin is very important. In MCQs they can write biotin, in MCQs they can write vitamin B7. So you should know also the names of vitamin along with their initials. Okay, now this is like a whole activation and transfer process. And then in the further steps they have like shown you that what happens next is uh, this pyruvate is added, okay, and then it becomes oxaloacetate and then malate. Okay, so this all thing which is going on is not like our business right now, but have to we have to focus in this reaction that what carboxylases are basically doing. Okay, this is your reactions of gluconeogenesis. Okay, when we go up to phenylephrine, okay, um, this is your pyruvate which is coming from your glycolysis. Okay, and this is your oxaloacetate. I told you in the very first class of biochemistry metabolism that pyruvate has 4 fates. So one of the fate is pyruvate is directly converted into oxaloacetate, and oxaloacetate is a product of Tca cycle 2. Okay, so if you're like mixing in your head, let me show you this picture from your textbook, first aid book. Okay, you see this picture over here. This pyruvate is the product of glycolysis cycle. Okay, and it is directly converted to oxaloacetate as well. Okay, it's not necessary that it will convert into acetyl CoA. If you remember, we studied that when the levels of NAD to NADH ratio is more in ethanol metabolism, Tca cycle do not work. If you remember this process, and I told you that pyruvate will be converted to oxaloacetate, and then oxaloacetate will be converted to malate. Okay, and Tca cycle will not work. The acetyl CoA that will be formed will be shifted to either lipid metabolism or either to ketogenesis. If you remember the picture, it's the picture from your first aid book. Okay, so the same thing's gonna happen over here is oxaloacetate is being converted to malate. Okay, and then this malate will, this is from the mitochondria, this malate will move to the cytosol. Okay, and then this malate will be converting to oxaloacetate again, and then phosphoenol pyruvate. Once phosphoenol pyruvate is formed, once PEP is formed, that PEP will go up back into where it will go back up to the gluconeogenesis cycle. So see here, this oxaloacetate can be redirected to the formation of PEP. Okay, and then this PEP will lead you to this gluconeogenesis cycle. So this is about the picture that they have drawn over here, but I showed you this picture only to make you understand what pyruvate carboxylase is doing and how is this reaction going on. Okay, so now if you're clear with this point from your book, now if you're clear with this carboxylase point from your book, let's see some enzymes that are rate limiting and has carboxylase in them. Okay, let's see some enzymes. Okay, so we are left with these enzymes, and let's see which enzyme has carboxylase. So in fatty acid synthesis, we use acetyl CoA carboxylase. We say it ACC. Okay, so let me show you this reaction of fatty acid synthesis from your first aid book. Okay, now you see this picture over here. This is your fatty acid metabolism. Okay, we only have to see synthesis. Don't see to degradation because the enzyme is different. Okay, when we see synthesis, you see this hormone, this enzyme name written over here, acetyl CoA carboxylase, ACC. Okay, so what's going on is this synthesis process goes from your mitochondria to your cytoplasm. Like the direction of arrow is like this. But when we talk about the degradation, the direction of arrow would be like this. Okay. So like we are only seeing this synthesis right now. So you see the citrate, from where the citrate comes from, citrate comes from Tca cycle. Okay. Now if I have to like draw a little Tca cycle for you, yesterday I showed you that we have this citrate, and then citrate is converted to isocitrate, and then isocitrate is converted to alpha ketoglutarate. Okay, if you remember, and then alpha ketoglutarate is converted to succinate. So you should learn these all steps. Okay, so let me open another picture from like your Google, so that we don't have to shift pages. Okay, see this. See this picture. You have this acetyl CoA, pyruvate is converted to acetyl CoA, and then this acetyl CoA along with oxaloacetate, and the pathway moves like this. Okay, so you see here, um, we have this citrate, we have this isocitrate, we have this this picture is not right. This is not right. Wait, yes, this is fine. Okay, now we have this citrate, isocitrate, alpha ketoglutarate, succinyl CoA, succinate, fumarate, malate, and oxaloacetate. So the things that you are seeing over here, this citrate, okay, this all Krebs cycle, this Tca cycle goes in the mitochondria. Okay, in the first lecture, I told you you have to remember all the energy cycles that are giving you ATPs and NADPs goes in the mitochondria. The Krebs cycle is going inside the mitochondria. Okay, what will happen? What will happen is that this citrate, okay, from your mitochondrial matrix will move to your membrane. Okay, and then it will be converted into acetyl CoA in the cell cytoplasm. Okay, so you see it is again converting into acetyl CoA. When you are learning this Tca cycle, you see acetyl CoA is converting into citrate, but in fatty acid synthesis, citrate again converts into acetyl CoA. Okay, and if I have to show you a picture of Hmm, it's not that interesting. This picture was much better than others. So let's see it in this picture. Okay, you see here now. This is your Tca cycle, and this is your mitochondria. They have drawn this whole thing is your cell, and this is your mitochondria. So this is your Tca cycle going on, and you see acetyl CoA was converting into citrate in your Tca cycle, but this citrate can move out from your mitochondria to your cell cytoplasm, okay, and it can enter into your fatty acid synthesis. Now they have not shown all these steps, but now you can actually get the idea of how citrate can be reconverted to acetyl CoA, and then basically that acetyl CoA enters into fatty acid synthesis. Okay, so as in written in your book. Now this is step where acetyl CoA is converting into malonyl CoA. This step requires acetyl CoA carboxylase. Okay, and now you have read what carboxylase enzyme was doing. It is transferring carbon dioxide from the biotin. So now you see, in even in your first aid book, they have written it so nicely. If you understand this thing back there, when you will study fatty acid metabolism, this picture will not be new for you. Okay, so that is why I'm also using the same pictures of your books too, so that you may understand it well. So now you're clear with from where the citrate comes. The citrate is coming from your Tca cycle. Okay, from the mitochondria it is converting to the acetyl CoA. Okay, and then in the acetyl CoA is converting into this malonyl CoA. So this step is basically like, wait, this is step only is your rate limiting step. Okay, fatty acid synthesis rate limiting step is this, and you have to remember the name of enzyme, acetyl CoA carboxylase. Please remember the name of enzyme, acetyl CoA carboxylase. Okay, now if I take you back again to the, okay, this was page number 89. If I take you back up to the book, now you're clear with this point that fatty acid synthesis has this enzyme. You have seen where it works. Okay, now if we see the regulators at this level, see, I have yesterday told you that the insulin and glucagon works opposite. Okay, glucagon is always secreted when we have this low level of glucose. Okay, and insulin is secreted when we have high level of glucose, because insulin controls our glucose levels in the blood and it lowers it down. Now the more glucose the more glucagon your body is secreting determines that your body has low glucose levels. Okay, you are having low glucose levels, that is why your body is secreting more and more glucagon, and you are in a fasting state. So you need to synthesize fatty acids. You need to synthesize fatty acids when you are in a fasting state. Okay, so please remember this point. You are in a fasting state. Now you need to synthesize fatty acids. What is the fatty acid, or you can say TAGs, your triglycerides? These are the shorter units of your fats, your huge fatty chains. Okay, you all might have studied regarding the 20, 30 carbon fatty chains. Okay, the if you have heard of the names like um, palmitoyl or arachidonic or others. Okay, so like these are like long chains, but when we break them down into smaller compounds, they are termed as either triglycerides or fatty acids. So when you're in a fasting state and your blood sugar level is low, the pancreas alpha cell secrete this glucagon hormone, okay, and that glucagon will inhibit this cycle. That glucagon, more glucagon will be secreted, and that glucagon will inhibit this fatty acid synthesis, because we are already in fasting state and our glucose levels are low. We do not need more fat breakdown, okay. We need it to shift to some gluconeogenesis, or we need it to shift to some at other levels. So in a fasting state, in order to raise glucose levels, this glucagon will inhibit this fatty acid synthesis, okay. But in a well-fed state, when your glucose levels are high, this insulin will be secreted, and this insulin will promote this fatty acid synthesis so that it may move towards this gluconeogenesis more and more. You're getting my point. So if I clear these all drawings, and if you just have a look on glycogenesis, fatty acid synthesis, in both of these things you see the same regulators. Why? Because the mechanism is going the same. We were yielding glycogen, okay. We were from small glucose units, we were making this huge chunk of glycogen. Why? Because our body is already in high in glucose, and we do not want these little chunks. We already have so many little chunks in our blood that we now want them to unite and store it as glycogen. So our body was more with glucose levels. Insulin was secreted from your pancreas. That insulin promoted the production of glycogen from this little glucose molecules. And the same thing is going at this level too, okay. The glucose levels are high, insulin is secreted from the pancreatic beta cells, and they will promote the synthesis or the formation of fatty acids for future purposes, okay. So in future, when you are like deprived of glucose, and your fats needs to be break down, and you want your fats to break down, and you want your fats to enter into the gluconeogenesis cycle. So for that future purpose, we are synthesizing these fatty acids. You're getting my point? So the mechanism of glycogenesis and the fatty acid synthesis is like different, but the root is same, okay. The purpose is same. You're getting my point? The purpose is same. Now you see citrate here, positive. So definitely the cycle was starting from citrate. When you already have citrates in high amount, the cycle will go to the end fatty acid synthesis production, okay. More citrate will push the cycle more towards the production of fatty acids. So it will promote your fatty acid synthesis. But more palmitoyl-CoA, because the end product that you are forming is palmitoyl, okay. The end product you are forming is palmitoyl-CoA. Your malonyl-CoA will be converted to palmitoyl-CoA, okay. It is a fatty acid chain. So if you have more and more palmitoyl-CoA, it will say, I don't want to be produced more, please stop this production. So this more quantity of palmitoyl-CoA will cause this feedback inhibition of fatty acid synthesis, and that is what they have written over here. More and more palmitoyl-CoA will cause feedback inhibition of fatty acid synthesis, because palmitoyl-CoA is the end product, okay, and citrate is the substrate. The cycle is beginning from the substrate. So more substrate will lead to more fatty acid production, but more product will lead to the inhibition or the stop of fatty acid synthesis, okay. So please remember these mechanisms. Now, as you understood regarding the carboxylase and its functions, and we have seen the regulators, the next enzyme I will take you to will be synthase or synthetase. So we have already seen synthase yesterday, okay, and then you also, and then like we studied phosphorylase actually, but on the contrary, I taught you synthase too. But you see synthase and then you see synthetase written here, synthetase 1 and synthetase 2. So we have these four enzymes, okay. The you can call synthase as synthetase 2 and you can call synthetase as synthase 2 because the function of both are same, okay. But you have to remember that wherever you see genesis written, the end enzyme name is synthase. Like glycogenesis is glycogen synthase, ketogenesis will be HMG-CoA synthase. Okay, and then you have the synthetase 1 and synthetase 2. Okay, and you see the name of enzymes are exactly same. We have this carbamoyl phosphate synthetase 1, carbamoyl phosphate synthetase 2. So the only difference lies in 1 and 2. And one of them is of de novo pyrimidine synthesis and the other one is of urea cycle. So even the processes are two different. Like this is pyrimidine and this is urea. Okay. So now like the way I remembered it was like this, that we always take glycolysis first, okay, and then there comes this Tca cycle, and from Tca cycle we have urea cycle. The fumarate of your Tca cycle enters into your urea cycle. Okay, so I took it this way that because we study this carbohydrate metabolism first, and soon after from Tca we are going into urea. So I learned it that the first comes first, you know. So when we talk about the earliest cycles we study, so the enzyme name 1. Okay, so you can also like make anything in your mind if you can like really getting it, so you can just learn it by any ways. So the main sole purpose is to remember where is synthetase 1 and where is synthetase 2, so that you may not make the mistake. There is a possibility that they might ask urea cycle rate limiting enzyme in MCQs more than pyrimidine synthesis, because urea cycle is more easier and more important. So they don't go in too much complexities as well, but they try to stick to the favorite and favorite topics of biochemistry. So now, if I take you to the functions of synthase or synthetase, so you see, they joins two molecules together using a source of energy. So now they require this synthase or synthetase requires energy, okay? That energy may be in the form of ATP, that energy may be in the form of acetyl-CoA. You know, coenzyme A cannot be formed without a energy molecule, okay? Or nucleotide sugar. What is a nucleotide sugar? We have seen, we have two words now. Let me take you to some basics. We have these two words, nucleoside and nucleotide. Okay, T, say remember try. Okay, from the word T, remember try, and the remaining one take it as bi. Okay, why? The reason is when we talk about nucleotide, we have three components. It is made up of three components. Okay, number one, a sugar. Number two, a phosphate group. And number 3, a nitrogenous base. Okay, remember this. From the T, remember tri. A sugar, a phosphate group, and a nitrogenous base. So here in the nucleotide sugar, they are talking about this sugar, which is attached with all these components to make a nucleotide. Okay, so if I show you a picture of a nucleotide, let's say, let's see a picture of a nucleotide. You see this picture. Where is the picture gone? You see this picture, how beautiful picture is this? This is a sugar, okay? This is a phosphate group and this is a nitrogenous base. So combiningly, we call this whole thing, three things a nucleotide, okay? So they are talking about this sugar, not any other sugar like glucose or something else. No, this sugar, specifically this sugar, and this sugar is not a glucose because glucose is not a pentagon. You see, this is a 5-carbon structure like 1, 2, 3, 4, and 5. This is a 5-carbon structure. This sugar is always a pentose sugar. So in a nucleotide, the sugar is always pentose. It is never a hexose, okay? And glucose is an hexose sugar. So glucose is, we are never talking about glucose here. So please remember this. These are very important points that I am telling you right now. The sugar should always be pentose, okay? And if I show you, just to make your concept good, a nucleoside image. So now you see the difference. This is such a good image. Okay, now you see the difference over here. What is the difference between a nucleoside and a nucleotide? You do not have you have a phosphate group in nucleotide and you do not have a phosphate group in nucleoside. Okay, so remember T stands for tri, so that you may remember we have 3 structures, okay? And this sugar molecule is 5 carbon, okay? 5 carbon, not 6 carbon. So let's go back to our book. And now in your book, you see these enzymes, these 2 enzymes, they join the 2 molecules together using a source of energy. And that source of energy can also be a nucleotide sugar, okay? So let's see which enzymes are these and how are they joining 2 molecules together, okay? Let's see it practically. Now, let me see in the book if I can see a synthase or a synthetase nearby, even in any other example. This is gluconeogenesis, okay? This is glycogen metabolism, okay? Here we will see okay, now see this image. This is glycogen synthesis, okay? This picture is of glycogen synthesis. And what is going on? This is your glycogen synthase, your rate limiting step and your rate limiting enzyme. And we just read the function of synthase as it joins 2 molecules together, okay? So this cycle is starting from your glucose 6-phosphate, the structure of your the component of your glycolysis, okay? So if you remember from the yesterday's lecture, we have this glucose and then this glucose was converting into glucose 6-phosphate, okay? And then this was converting into fructose 6-phosphate. But this glucose 6-phosphate was also converting from glucose 1-phosphate, and this glucose 1-phosphate is being converted by glycogen. Okay, we have read a pathway like this also. So if I take you to this image now, you can clearly see that glucose 1-phosphate can be converted to glucose 6-phosphate, and then 6-phosphate can interconverted into 1-phosphate. So there's a double arrow. Okay, so here is from where your glycogen pathway is starting. Okay, and then you see at this particular level, this glucose 1-phosphate becomes UDP glucose. So this thing, what is a UDP or what is a UTP? It is same like ADP or ATP. We have this nucleotides, adenine, guanine. These are the nitrogenous bases. Okay, adenine, guanine, cytosine, thymine, and uracil. So when we say ATP, it is adenine triphosphate. When we say GTP, it is guanine triphosphate. When I'm saying UTP, it is uracil triphosphate. So this is not a new thing. Okay, it is the same thing. They are 5 nitrogenous bases, and you can change it to ATP, GTP, UTP, CTP, TTP. Okay, anything. So here we are taking UTP, like uracil triphosphate, and this UTP joins with this glucose 1-phosphate to make this UDP glucose. But now you see what is happening at this particular level, that this UDP glucose, okay, is being converting into this long chain, because you are making a glycogen. A glycogen is a polymer. Okay, a sugar like glucose is a monomer. Okay, so now you are making a polymer, a long chain. So you see this UDP glucose, let's say this red color thing is a glucose. Okay, so you join multiple structures of glucose. Okay, you join multiple structures of glucose. So basically your enzyme glycogen synthase is doing the same thing that is written in your book, the same function, that it is joining 2 molecules. So now you see this was one molecule, this one red dot is number 1, and this another red dot is number 2. What are they? They are glucose molecules. They are glucose molecules. So you are joining you're joining 2 molecules together via an enzyme synthase, and then this synthase is joining this happens in like multiple steps. It's not like in one step, multiple steps. So 2, 2, 4, 4, 8, then 12, then 16. So this is how you make this big huge chain. So this is like a very easy step. But they said that synthase or synthetase joins 2 molecules in the presence of an energy compound. So can I say that UDP is an energy compound? Yes, I can say that, because when ATP can gives you energy, when GTP can gives you energy, why not UTP? Why not UTP? UTP is uracil is also a nitrogenous base as adenine and guanine. So if ATP is an energy compound, GTP is an energy compound, UTP is also an energy compound. Okay, you get my point? So synthase is joining 2 molecules with the help of an energy compound. So now this UTP and this UDP, they all are energy compounds, and it's joining 2 molecules together. Okay, and then you see it makes this whole big chain by joining molecules. Okay, so people, like this is a very good example of and this is also your rate limiting step. Okay, so you just see in your first state that this glycogenesis, you just see the cycles of glycogenesis and this function of this synthase enzyme, what it's doing. Okay, so if we see another example of let's say Let's see some other example. Let's see an example of synthetase now. I have to like search a bit more because I don't know the number of chapters. Or I may search it on I may search this cycle on Google for you people. What I was searching for the de novo pyrimidine synthesis. Okay, so let's see a picture here. Hmm. Okay, this is purine synthesis, not pyrimidine synthesis, but I just want to see a synthetase enzyme. So they have shown an enzyme, but okay, now finally I can see something. Are you able to see this picture? Okay, so now you can see this picture on your screen. This step, this first step is using your carbamyl phosphate synthetase 2. Okay, this first step is using this enzyme. If I can, if there is like a more clear picture. No, sorry people. We have to stick focused on like this picture because it's very difficult to find this de novo synthesis. Okay, um, so I'll not waste time. I just want you to show that at this enzyme, carbamyl phosphate synthetase, okay, is like it is because this is pyrimidine synthesis. Okay, so in pyrimidines, like we have purines and we have pyrimidines. There's a difference in between their structure. One is like a two-ring structure and the other one is a one-ring structure. You're getting my point? So this is the difference between the pyrimidine and purines are named because of the differences in their structures. And if and like here is a mnemonic which I want you to remember. The mnemonic is pure as gold. Okay, please remember this mnemonic, pure as gold. Remember the mnemonic pure as gold. What is pure as gold? Let me tell you. Pure as gold. So A is adenine and G is guanine, okay? And P is purine. So we have this 5A, G, C, T, and U. We have these 5 nitrogenous bases. Among 5, this A and G are purines and C, T, U are pyrimidines, okay? So when we are saying that we are dealing with de novo pyrimidine synthesis, we are saying we are synthesizing cytosine, thymine, and uracil. So now even in this picture, if I rub this, clear this all, and I zoom this picture for you, see this cycle. In this cycle, you can easily see UDP, UTP, CTP, okay? So they have, like in this cycle, they have shown uracil and cytosine, but thymine is also in here, okay? So, and the very first step of this cycle has this synthetase 2 enzyme, okay? So this is like this synthetase 2 enzyme is of de novo pyrimidine synthesis. But if I search urea cycle for you here, now urea cycle will be easier to find. Now if I search this urea cycle to you, very good picture, a really good picture of urea cycle is this one. So see this picture now of urea cycle. This is mitochondria and this is cell cytoplasm, okay? And see this enzymes here. One enzyme is this is your synthetase enzyme, but this is not your rate limiting enzyme. Your rate limiting enzyme is this one, carbomoyl phosphate, sorry, this one, carbomoyl phosphate synthetase 1. This is your rate limiting enzyme, okay? So this urea cycle is starting with, this is your Tca cycle, okay? From your Tca cycle, this is your fumarate, okay? The fumarate from your urea cycle enters into your Tca cycle. This is asked in your MCQs previously also, so remember, I'll show you in your book also. And your cycle, it starts with ammonia, amino acid metabolism. And you see this carbomoyl phosphate synthetase 1, okay? This is your enzyme which is using this high energy molecule ATP and combining and joining 2 molecules together. This ammonia and the other one is not clear in this picture because they have like cropped it or something, but let's see it in this picture. Let's see it in this picture. In this picture also you can see now, this is your mitochondria and this is your cell cytoplasm, okay? A very good picture. This is your ammonia, okay, which is coming from proteins catabolism, okay? And this is bicarbonate. So your ammonia and bicarbonate is joined together, okay? The enzyme is carbomoyl phosphate synthetase 1, CPS1, and it joins 2 molecules with the help of a high energy compound, that is ATP. So the function of synthase and synthetase is exactly the same, okay? And you are supposed to remember these rate limiting enzymes, okay? Don't like forget the reactions or something. I'm only showing you to explain it, but remember the rate limiting enzymes of every cycle. Also please remember in this urea cycle that this fumarate will enter into your Tca cycle, okay? This fumarate will enter into your Tca cycle. So this is very important step, fumarate, okay? This has been asked previously in your MCQs, so you have to remember this. Now I'll take you back to the book. Okay, so in your book now you see urea cycle carbamoyl phosphate synthetase 1. Okay, de novo pyrimidine synthesis carbamoyl phosphate synthetase 2. So remember this 1 and 2. Okay, and then glycogenesis with glycogen synthase and ketogenesis with HMG CoA synthase. I'm not showing you ketogenesis picture. It is in your first aid. Okay, so we will be seeing it when we will be seeing both of these together. Okay, both of these together we will see that. So for now, I just showed you that this pyrimidine synthesis, at the end of this pyrimidine synthesis you will be making UTP. So if you have more UTP, this UTP will inhibit this cycle, feedback inhibition. Okay, the product is more because the product of pyrimidine synthesis, last product is uracil triphosphate. You already have uracil triphosphate. Do you need to synthesize it more? No. So it will cause a feedback inhibition. Okay, but ATP will promote the step, PRPP will promote the step. Okay, why these two things will promote the step? Because PRPP is the substrate, okay, and ATP is a low energy molecule as compared to UTP. UTP is a more higher energy molecule, okay, and ATP is a lower energy molecule. So we are using energy because this is synthetase. It is using an energy molecule to go to the next step. Okay, so it will use these purines, nitrogenous bases, energy molecules to create a pyrimidine nitrogenous base energy molecule. Okay, you should get my point now. So UTP, TTP, and CTP, these are your pyrimidines. Okay, for purines we have A and G, like, okay, A and G. So now if you have a look on the regulators of your purines, you will see that I told you A and G. So you see GMP, this will be a product, so it will inhibit this cycle. But A is also present over there, but this is not ATP, this is AMP. If we see the range of energy, AMP has less energy as compared to ATP. So if you have ATP, or if you have GMP, or if you have GTP, this will inhibit this purine synthesis cycle. But if you have AMP, this will promote this purine synthesis cycle. Okay, so these things are very important. And then inosine monophosphate is some other thing. Okay, so we will see it when we will be studying this in more detail. Okay, now you are not usually asked all the regulators in your exams. You have to remember glycolysis regulators, especially fructose 2,6-bisphosphate. Okay, please remember the Tca cycle, glycogenesis, and glycogenolysis regulators. Okay, and remember N-acetyl glutamate in urea cycle. So if you will remember these regulators, that are more than enough for your MCQs, because they don't go in so much complexity. They ask the common ones, okay, the easier ones. So you have to remember like these. Okay, so don't panic for urea cycle and acetyl glutamate, because it is the substrate. Okay, it is the substrate. So we will begin the cycle from that level. So I will teach you in the details of urea cycle. Okay, so now we are left with HMP shunt, okay, and ketogenesis, cholesterol synthesis. Okay, so the enzymes for ketogenesis and cholesterol synthesis are same. For genesis you have to remember synthase, but for cholesterol synthesis the enzyme name is reductase. So let's see a bit from your first aid book itself. If the lecture would stop, please join again. Okay, I will paste the link. So see this, first of all, see this urea cycle picture. This is your N-acetyl glutamate. This is your substrate, okay? So if this N-acetyl glutamate will be more, it will promote this cycle, okay? So this is like a very simple thing you have to remember, okay? But if I'll show you the cholesterol synthesis, so even in your first states, you have this diagrams of cycles, so you can see it from there also. So this is a cycle, but they have not written the name of enzyme over here. This is your ketogenesis, okay, ketone body formation. But the name of enzymes are not mentioned here, okay. It's just written in the description that you have this HMG-CoA reductase for cholesterol synthesis and HMG-CoA lyase you can say, or HMG-CoA synthase you can say, for your ketogenesis or ketone production. So they have not mentioned the enzymes at which step they are being used, but they have like kind of showed you the mechanisms, okay. This is the step where HMG-CoA is converting to acetoacetate. So this step is probably the step that uses this HMG-CoA lyase or HMG-CoA synthase, okay, people? Because we have like seen in previous enzymes that the enzyme name also comes from the name of substrates, okay? So this is the step where you are using this HMG-CoA synthase or lyase for ketogenesis. And these three are your ketone bodies. We have three ketone bodies, okay? So these are the three ketone bodies. You should know the names of ketone bodies: acetone, acetoacetate, and beta hydroxybutyrate. You should learn the names of ketone bodies, okay? And then so this is like the last things of your metabolism, like last chapters, okay? Yes. So like let's go back to our portion. Like we have quite completed the metabolism thing now, okay? We are left with this HMP shunt, and I will show you this very important enzyme G6PD. Just you only have to remember the name of enzyme with HMP shunt, okay? Very important it is, G6PD. So this is your basically HMP electron transport chain. So you might see the enzyme here if possible. Like I'm still not sure. It's easier to search on Google. Let's search it on Google, so you might be clear, because HMP shunt is important, okay? So this is your hexose monophosphate shunt, and this is your step. You're using this enzyme at this particular step. This is a very good picture, okay? This is a very good picture, and you see it is also starting from your glucose, a hexose sugar, okay, and then glucose 6 phosphate is being converted to your 6 phosphogluconate and so on, okay, and then this goes to your nucleotide synthesis, okay. So at this particular step you are using this dehydrogenase enzyme. If you remember yesterday, I told you that dehydrogenases are the enzymes that are involved in redox reactions. So the redox reactions are the reactions which gain or lose a proton, okay, a H positive. So you see the same thing you are seeing with this dehydrogenase enzyme as you have seen in the yesterday ones, okay. I showed you the Tca cycle dehydrogenase enzyme. That was isocitrate dehydrogenase, the rate limiting step of Tca cycle. Similarly, I showed you pyruvate dehydrogenase enzyme too. So today we are seeing another example of a dehydrogenase, okay, which is a rate limiting enzyme of your HMP shunt G6PD. Please remember the name G6PD. Okay, you don't need to remember the reactions or anything. You only need to remember the rate limiting enzymes with the name of your HMP pathways. And you will be studying this G6PD deficiency in your hematology. Okay, this is the most common thing they do ask in pathology. Heme bodies. G6PD deficiency, heme bodies are seen in your blood. Okay, so just because it is written over here, so I'm like kind of reminding you. So this is like one of your revision too. Okay, so now let's go back to our first aid. Okay, so we are finally done with our enzymes, and there were two enzymes that were left, mutase and hydroxylase. So the name hydroxylase is just telling you to add a hydroxyl group, simple as that. So tyrosine hydroxylase we have studied already in our previous pharmacology. We have studied that from tyrosine you produce dopamine, epinephrine, norepinephrine, right? So you are adding a hydroxyl group to that chain and you're forming new compounds. So it's simple, adding a hydroxyl group. So when we talk about mutase, you see it relocates a functional group within a molecule. So it's like changing the positions. Okay, it is causing mutation. So they have given the example of vitamin B12 dependent methyl malonyl CoA mutation. Okay, so wait. So this is your picture. This is your enzyme, methyl malonyl CoA mutase, okay? And this is B12. This step is B12 dependent. I've explained you this full picture in very much detail in B12. And I also told you there are 2 to 3 MCQs from this picture. So please, if you have not listened the B12 lecture, please go and listen it back. Okay, so this is your step of methyl malonyl CoA, which is converting into the succinyl CoA, and the enzyme is mutase. So basically what this enzyme is doing, it's just changing the position of the molecules that are present in these structures. Okay, so this is also your example of first aid book. So you need to remember the name of the enzymes that are used, the vitamins that are used for the cofactors of these enzymes, like B7 for carboxylase, okay, B1 for your dehydrogenases, okay, and mutase B12, okay. So please remember these names of enzymes along with your vitamins. So this is like very important thing. And then I have taught you all the rate limiting enzymes. So I think you're very much clear with the pictures and the processes and everything now. And you might have a little, if you can like go through the recorded lecture also once again, and you can take screenshots of those Lippincott images for your revision. Okay, so that it may help you while you're revising it finally. And now, before ending the lecture, I just want you to see this picture, a very closer look to this picture. This is the most beautiful image of your metabolism. All the all the pathways are present over here. Okay, even they have shown you the symbols where your biotin like B7 cofactors are using and your TPP is using. Okay, so they have even shown you, and there is this key for your enzymes. Okay, and the numbers given. So you can see, now you see this HMP shunt over here. It was starting, we saw this picture of HMP shunt, and it was starting from this glucose. Okay, so in your book also you see this HMP shunt is starting from your glucose. Okay, it is starting from your glucose. Okay, and then glucose 6 phosphate, and then it moves to your uh, either your mitochondria or your cytoplasm, because HMP shunt takes place in both. Okay, so we have learned hugs, mnemonic, and HMP shunt was one of them. Okay, so remember that too. And then it just have these two steps. Okay, so this is step where glucose 6 phosphate is converting to 5 phospho gluconolactone. Okay, so this number 5 is G6PD enzyme. So this is your rate limiting step. Okay, this is your rate limiting step. So they have, this picture is also very good. I like this because you see there is this numbers which do not have a red lining over there, but there are these numbers which have a red lining over there. So all of these red linings are the rate limiting enzymes. So you can also learn them from here. Okay, so this is a very good picture, and you can see how these all cycles are interconnected with each other. Even the urea cycle is given over here, and this is your carbamyl phosphate, and this step uses the enzyme carbamyl phosphate synthetase 1. Okay, 1. Please remember urea cycle 1. And you also get that N-acetyl glutamate from here, which is your substrate. So, and I was telling you about fumarate. So you see, when arginosuccinate is converted to arginine, the fumarate is released and it goes to your Tca cycle. So there are so many things you need to remember for Tca cycle, but we will study it when it will be the turn of Tca cycle. Okay. So for now, we have done with that, and we have even correlated this picture every time. So now ATP, if I talk about ATP, so you know there is a bit difference in your lipin part, in your Kadzang, and in your first state in terms of ATP. So the best way to remember, in here they have shown you, if you summarize this whole diagram, okay, they are telling you that you are producing 32 net ATP via this malate aspartate shuttle. Okay, malate aspartate shuttle is a shuttle of your urea, of your Tca and your gluconeogenesis. Okay, so 32 net ATP by there, and then 30 net ATP via glycerol 3 phosphate shuttle, which is the lipid metabolism thing that is going on in your muscle. Let me show you up in the diagram. And then from glycolysis you are only getting 2 net ATP. Okay, and that's it. They have not told you about the ATPs that you are producing from your Tca cycle. So please remember, from the Tca cycle you are getting 12 ATPs in total. Okay, so you are getting 3 NADP. Okay, 3 NADP means 3 threes are 9. Okay, 9 ATP, because 1 NADP is equals to 3 ATPs. Okay, so 3 NADPs means 9 ATPs. Then you have a GTP. So GTP gives you 1 ATP, and then you have an FAD, and FAD gives you 2 ATPs. So you get a total of 12 ATPs by 1 acetyl-CoA. But in your Tca cycle, you do not have 1, you have 2 acetyl-CoAs. So in total, you will be getting 24 ATPs. Okay, please remember this from your Tca cycle. And then 2 net ATPs from your glycolysis. Okay, but this is like if you do the math, the two net ATPs that you're getting from your ATPs, you are utilizing that in TCA cycle. So we will see it in a closer look when we will study TCA. Okay, but from while here, if you see, this is your malate aspartate shuttle. Okay, so your urea and your TCA cycle, and then it goes back up to your gluconeogenesis. So this is one thing, and the other one is your glycerol and phosphate, but it is not shown in over here. It is not shown in this picture, but we will like tomorrow we will see it in the picture of your Lippincott, page number 92. Okay, so that picture is more good and clear and better because that's only of carbohydrate. Okay, more. So tomorrow we will begin our lecture, because if I'll go in detail, then I have to open the cycles to let you explain at which step which energy compound is using, and it will take another long time. So we will see this thing tomorrow, and we will also see the differences of these hexo and glucokinase, along with these glycolysis cycles and TCA cycles. And I have taught you this 4 fates of pyruvate already. So we will see these things in detail, and inshallah ta'ala I will try to cover your TCA cycle tomorrow. There is a lot to learn from this picture, a lot of things. Okay, so today our biochemistry lecture is just ended, and our next lecture will be of pharmacology. Probably it will be at 1:45 or at 2 o'clock. So we will be meeting after Zohar prayers. Okay, people? So this is like all about today's lecture. Do study. Okay?